Hello my dear friends, welcome to Live Prep. In the previous lecture, we studied about some introduction related to biomolecules, and there we observed that when we are comparing both living and non-living things, so in both of these, the elements name purpose, if you are observing, the list of elements or the list of these particular elements present in living and non-living remains the same, but proportion wise if we see, there is a difference. This is what the information we have studied. And now we are going directly with the living tissues. In the living tissues, what are the elements that are present? What are the compounds that are present? How exactly we are performing an experiment? In the NCERT, very clearly they have given the complete procedure, and the same thing in the form of a diagram I will try to explain you. Slowly we are going into this particular concept. Clear? And please remember, once again I am telling you, whatever the information we are studying here, this is actually a continuous information, but in the NCERT, in different pages, in different paragraphs, they have given this, and the entire information we are combining together for proper understanding. I hope now you can understand this. And now we are going with how to analyze the chemical composition for a living tissue. In the living tissue, what are the various compounds that are present, what are various elements that are present, how exactly we are performing experiment, everything I will tell you. But please remember, chemical experiments we are not studying here. Simply the procedure in simple, in general, we are trying to understand. That's it. Okay. Anyhow, this sentence I will read at the end. First, let me complete the entire information. And at the end, we will come and then simply we will read this. You will understand each and every point of NCERT. Clear? And now let us quickly go through this. And now we are going with a living tissue. First, let us take a living tissue. Living tissue. In the previous lecture itself, I told you what are the various examples that you can take for the living tissue. You can take a piece of plant and it is considered as a plant tissue, and you can take a piece of an animal, you can take that as animal tissue. Anything you can take, or even you can take some group of cells for the microbes and you can take this as a microbial paste. Microbial paste. Anything you can take. You can take either this, anything you can take, no problem. Yes, and you are ready with any of your choice, and let us take any of this. For our easy understanding purpose, let us take an animal tissue. Okay, let us take an animal tissue. And where we are taking this, I will try to draw some diagrams so that when we are going through NCERT content, you can easily interlink this. First, let us take this particular one in a plate, or in a small beaker also you can take this. Suppose this is the tissue. This is the tissue. And how exactly we are proceeding with this? To this tissue, we are adding a particular solvent. What is that solvent? Observe this. The solvent we are adding is trichloroacetic acid. What is this trichloroacetic acid? First, we forget about trichloro. What is acetic acid? CH3COOH. CH3, 3 H's are present. CH3 COOH is called as acetic acid. In this, instead of these 3 hydrogens, if 3 chlorines are present here, that means here chlorine is there, here also chlorine is there, here also chlorine is there. Now it is called as a trichloroacetic acid. Already you know that chlorine is electronegative element. Yes or no? Chlorine is already you studied in that periodic table, right? It is an electronegative element. So because of this, it has the tendency to pull the electrons towards this. For this shared pair of electrons, it is pulling this one. So that's why what happens, it will get partial negative charge, this will get partial positive, same way like this it is there. That means in simple, if you say there is some polarity here, there is some polarity. So that's why trichloroacetic acid is a polar solvent. Please remember this sentence. In this lecture itself, when we are specifically talking about the lipids, at that time I will use this point once again. So please remember here. So we are removing this. So what is the simple conclusion? So we are taking a tissue in a beaker or in a plate, and to this we are adding a solvent. What is this solvent? Trichloroacetic acid. This is a polar solvent. It is a polar solvent. This is the most important point you need to remember. Trichloroacetic acid is a polar solvent. It is water soluble solvent. So please understand this, polar solvent, and that we are adding here. So we have added this, and then with the help of mortar and pestle you can grind it, or else nowadays we are having some homogenizer. So that homogenizer will also do this mixing only. So because of this vigorous mixing, what happens? This will be broken into pieces. Now what happens? This tissue is broken into pieces, and then what happens? Cells also will break. Now what happens? Cellular content, that means the components present inside the cell are released into this particular solvent. Now this is how you are doing this. I will repeat again. Initially we are taking a tissue in a small beaker, and then we are adding trichloroacetic acid, which is a polar solvent, and then we are homogenizing, or we are breaking this. In simple term, if you want to say, breaking. So generally we are breaking this in a mortar and pestle. There will be a mortar and there will be a pestle like this, and with the help of this you can grind it, so that ultimately the cells will be broken. What is the ultimate conclusion? Cells will be broken and the cellular content is released into this particular solvent. So in this way, overall, if you see what happens, now here you will observe one slurry. So that now what happens? You will observe a slurry. Slurry means what? Semi-solid paste. Semi-solid paste. Okay. So that slurry you are getting here. Now this slurry is ready. And then what we are going with this? And then what we are doing is, observe this diagram carefully. We will take another fresh beaker. We will take another fresh beaker. On this beaker, we are keeping a cheesecloth. We are keeping a cheesecloth on this. You can use cheesecloth. You can use a cheesecloth, or you can even use cotton also. Please remember, anyone you can use. You can use cheesecloth or you can use cotton. Before directly going into the concept, first I will tell you what is the significance of this particular cheesecloth or cotton. Let us see. For our understanding purpose, let us take a cloth. If you are observing this cloth, let us take this is a cloth. You just keep it against the light. Then what you will observe? You can clearly see that in the cloth there are some threads like this. In the cloth there are some threads like this, and there are some threads like this. Yes or no? So if you are taking the cotton, in the cotton also cotton fibers are present like this only. So that what ultimately we need to observe is whether it is a cloth or a cotton, and if you are clearly observing, it will have some pores. It will have some pores. Please observe this one. Pores are present. So that cloth you are keeping here. And then what we are doing? Already slurry is ready in another beaker. Now take this slurry. And then what we need to do? And then you just pour this. You just pour this slurry in this. This is the slurry that you are pouring on this, and then you are squeezing it. So in the NCERT they are using a word called a strain. Straining means what? You just squeeze it. Now what happens? In this cloth, this type of pores are present. Now what happens? In this slurry, there are different types of compounds are present. Yes or no? In the slurry, different types of compounds are present. Now, do all these compounds have same size? No. In this slurry, different compounds are present, and these compounds have different sizes. Please remember this, different sizes. Some are small, some are large like this. And when you are passing this slurry through this particular cheesecloth or cotton, then what happens? In the cheesecloth or cotton, there are some pores. Pores are having a particular size. Now what happens? Those molecules which are smaller than this pore. Understand this. Suppose these are the pores. Now what happens? Small molecules can easily pass through the pore. Understood or not? Suppose they are large molecules, they cannot pass through. They will be present above the cheesecloth only. So what happens? Now since this trichloroacetic acid is a liquid, that liquid will come here. Trichloroacetic acid will come here. So simply I will write this trichloroacetic acid as a TCA. trichloroacetic acid. This is the standard abbreviation, trichloroacetic acid, okay, TCA. And now, since it is a liquid, it can easily pass through this one. And please remember, when this liquid is passing through this, then what this liquid is doing? Those molecules which are smaller than this pore size of the cheesecloth, and those compounds, let me use a different color here. Some compounds are also coming along with the acid. Please observe this. Entire acid is collected inside the beaker. Along with this acid, small molecules. What are the small molecules? Smaller than this pore. Smaller than the pore of this particular cloth, they are also coming along with the acid. And there are some compounds that are large in size. They are large in size. Large molecules cannot pass through this. So that's why those which cannot pass through this, they are present above this particular cheesecloth. Understood or not? So in this way, once again I am repeating, observe carefully. We took the tissue in a beaker, added a trichloroacetic acid, and then we have ground it. Because of this, tissue is broken, cells are broken, content of the cell is released into this particular acid. And then it is forming a slurry. And that slurry is strained through cheesecloth or the cotton. And when it is strained through this, the acid is collected inside the beaker. Along with the acid, some compounds are coming into this. And then some molecules which are large in size, they are not passing through the cheesecloth, so they are present above the cheesecloth. Understood or not? So please observe carefully. In this way, what we are getting? Two fractions. A fraction that is collected inside the beaker and a fraction that is present above the cheesecloth. In this way, you are getting actually two components. Two components we are getting. And what are these names? Observe carefully. Names we are writing very clearly, so observe carefully. And now this content, acid plus some other components, all these things are collected. They have got filtered through the cloth. They have got filtered through the cloth and collected inside the beaker. And this is called as a filtrate. Filtrate. These are the technical names clearly used in NCERT. Observe carefully. Filtrate, filtered through the cloth and collected inside the beaker, and it is called filtrate. And there is another fraction. That fraction is not passing through the cloth, so that's why it is present above the cheesecloth. And since it got retained on the top of the cloth, it is called retentate. Retentate. It got filtered through the cloth, so filtrate. It got retained on the top of the cloth, that's why it is retentate. So two names very clearly used in NCERT. So these are the two fractions you are getting. And what this filtrate is, and these are all coming along with the acid, That's why this particular fraction is called as acid soluble pool or acid soluble fraction. You can also use the word fraction. Acid soluble pool or acid soluble fraction. That is the filtrate. And what about this retentate? It is not coming along with the acid. Please remember this. It is not coming along with the acid. It is present above the cloth. So that's why this retentate is called as acid insoluble pool or acid insoluble fraction, or simply it is called as a retentate. So please observe this content. So two fractions we are getting. One is filtrate, also called as acid soluble pool, and other one is called as acid insoluble pool or retentate. In this way, two fractions we are getting. Next point, point by point. All the points I am adding here only, so that when we are reading NCERT, then it will be easy. Now the next point is, what are present in this particular filtrate or acid soluble pool? Large molecules are present or small molecules are present? See here, through the cheesecloth, through the pores they are coming. So already pore is small. So who will come into this? Those molecules smaller than the pore, only they can come here. That means this filtrate or acid soluble pool contains small molecules. contains small molecules. Small molecules. That's why they are passing through the cloth. They are coming into this. What about the acid insoluble pole? It will contain large molecules. Large molecules. That's why they are not passing through the cheesecloth. Simple logic. They are coming into this beaker because they are small molecules. They are not going into the beaker. They are present above the cheesecloth because they are large molecules. For the large, technical word is macro, M-A-C-R-O, macro. For the small, the technical word is micro, M-I-C-R-O, micro. Now we are writing this. Contains small molecules. That means these are micromolecules. These are micromolecules. That means these are macromolecules. These are macromolecules. Macromolecules. Understood or not? These are the names they have given. Observe carefully. And where we are observing these micromolecules? From the living tissue. From the living tissue. That's why they are bio micromolecules. From the living tissue we are getting these small molecules. That's why they are bio micromolecules. Same way here also. They are obtained from living tissue. Large molecules present in living tissue. That's why they are bio macromolecules. This is what they are saying. This is another important point we need to observe. These are small molecules means what is the size of this? What is the average size of this? Average size they have given that they are having the size range between 18 to 800. So you can say this as 800 or you can simply say that less than 1000. Less than 1000. And what are the units? They are Daltons. Capital D, small a. Capital D, small a. So what is the size of the molecules that are present in the filtrate or acid soluble pool? They are having the size range between 18 to 800 Daltons, or simply you can say that they are less than 1000 Daltons. These are less than 1000 Daltons. And in the biomacromolecules, generally their size is more than 10,000 Daltons. 10,000 Daltons. You can also say this as more than 10 kilo, small k. Many students are making mistake while writing kilo. They are writing capital K. It is not capital K. It is small k. Small k, capital D, small a. Kilo Daltons. Kilo means what? 1000. 10 into 1000 means 10,000 Daltons. So you can write more than 10,000 Daltons, or simply you can write more than 10 kilo Daltons. Both of them are same only. So in this way, generally in this particular retentate or acid insoluble pool, majority of these molecules are having size more than 10,000 Daltons. And here acid soluble pool is having the size less than 1000 Daltons, generally in the range of 18 to 800. And with respect to 18, if they ask, what is this 18? It is the water. It is the water. For the oxygen, it is 16. For the hydrogen, it is 1. Likewise, how many hydrogens are there? 2 hydrogens are there. So 2 into 1, 2 plus 16, it is 18. Okay. So in this way, this is the smallest molecule that they are talking here. So in this way, 18 to 800 Daltons. This is what the size that they are present in the filtrate or the acid soluble pool. Clear? This is another information that we need to understand. And the next information we need to observe is what type of compounds are present in filtrate, what type of compounds are present in retentate. This is also another important point. Now this filtrate has both organic compounds. Filtrate has both organic compounds and inorganic compounds. It has inorganic compounds also. It has both organic and inorganic compounds. This is a very important point. Many students are making mistake here. So observe carefully. Acid soluble pool has both organic and inorganic compounds. But what about the acid insoluble pool? It has only what? Organic compounds. It has only organic compounds. It does not contain inorganic compounds. Many students are making mistake here. So observe carefully. So all the inorganic compounds will come into acid soluble pool because they are small molecules. In the case of living tissues, Inorganic macromolecules are not present. Inorganic macromolecules are not present. All the inorganic compounds in the living tissues are small molecules. That is the reason why we are getting all these things in the acid soluble pool. Even the ions also, inorganic ions are there, Na plus, K plus, all these. They will also come along with this acid soluble pool only. So that's why this retentate or acid insoluble pool has only organic compounds. Only organic compounds. Understood or not? Next one. Next, what are these organic compounds? What are these organic compounds that are present in this acid insoluble pool? There are 4 types of compounds are present. 4 types of organic compounds are present in retentate or acid insoluble pool. What are these? They are nucleic acids. They are nucleic acids, proteins, polysaccharides, and lipids. Please observe carefully. So these are the 4 types of organic compounds that are present in acid insoluble pool or retentate. I will repeat once again. 4 types of organic compounds are present. All are organic compounds only. Inorganic compounds are not present. And what are these 4 organic compounds? Nucleic acids, proteins, polysaccharides, and lipids. That's it. Now come to this acid soluble pool. In this, what are the organic compounds that are present? Some simple examples I will write. Nucleotides, nucleosides, even the nitrogenous bases, and not only this, some simple sugars are present. Some monosaccharide, disaccharide, likewise, some simple, small sugars are also present. Not only this, amino acids are present. Not only this, there are many other types of small organic compounds are present. All these are present. So in this way, various types of organic compounds are present in this small acid soluble pool. But all these are small molecules. These are also organic, but they are large molecules. Please remember, organic compounds. In the organic compounds also, if you are observing, so different types of phosphates are present. Different types of phosphates are present. Carbonates are present, etc. Likewise, they are present. Not only this, some ions are also present. Ions like Ca plus 2, Mg plus 2, Na plus K plus Cl minus, likewise many are present. So all these are inorganic type. Yes or no? But ions are not considered as compounds, they are simply ions. So in this way, if you are observing this is what the overall composition. So if you are taking a living tissue is having both organic and inorganic. All the inorganic compounds are coming in acid soluble pool. But within the organic, small organic molecules are coming in acid soluble pool. Large organic molecules are coming in acid insoluble pool. This is what they are saying. Is this clear? I hope now you understood this entire information. Clear? Let me quickly give this information. The content that is passing through the cheesecloth is called as filtrate or acid soluble pool, and this is having small molecules of size range 18 to 800 Daltons, or in simple less than 1000 Daltons, and these are simply called as biomicromolecules, and these are including both organic and inorganic compounds. In the organic compounds, some small molecules like nucleotides, nucleosides, nitrogenous bases, sugars, amino acids. Not only this, various other types of compounds are present. In the inorganic compound, different types of phosphates, carbonates, many are present. Not only this, even some ions are also present in this acid soluble pool. Now let us see it. Some molecules, due to their large size, they cannot pass through the pores of this cheesecloth. That's why they are present above the cheesecloth, and this particular fraction is called as a retentate or acid insoluble pool, and this is generally having the large molecules. That's why these are called as biomacromolecules. Generally, they are having the size of more than 10,000. Who will have this more than 10,000? These first 3. What are these? Nucleic acids, proteins, polysaccharides. So now I will go into the next concept. So now I am linking this. And actually these three are called as true macromolecules. These are true macromolecules. Why they are true macromolecules? Because these are originally polymers made up of many units. I will tell you that content. So that's why they are true. But lipids are not true macromolecules. And now I will go into this particular information. So regarding this, I will write separately. In the next slide, I will write. I hope now you understood this basic information. Clear? Now we are focusing on this macromolecular fraction. In the macromolecular fraction, that is also called as a retentate. Retentate, it has four types of components. It has four types of components. They are nucleic acids, proteins, nucleic acids, proteins, polysaccharides, and then lipids. Polysaccharides and then lipids. These are the four types of organic compounds that are present in the retentate or acid insoluble pool. And now in this nucleic acids, polymers, proteins, polymers, polysaccharides, polymers. But lipids are not polymers. Please observe this concept carefully. Lipids are not polymers. First, I will tell you what is polymer, and in this case, what they are actually made up of. First, we will see what is the meaning of polymer. What is the meaning of polymer? Poly means what? Many. Poly means many. Mer means unit. Polymer means what? Many units. That means these are made up of many units. Let us say simple example. Suppose this is one unit, second unit, third unit, fourth unit, fifth unit. Likewise, many are present. Since many are present, now combinedly this is called as one molecule. This is called as one molecule. Please observe carefully. So each unit is called monomer. Monomer means what? Single. Mono means what? Single. Mer means unit. Single unit. Single unit is called monomer. Likewise, many units will attach and they will form polymer. Understood? So in this way, many units will attach and they will form a polymer. So in this way, in different polymers, different monomers are there. Suppose this is a nucleic acid. Suppose this is entirely nucleic acid. Now what each unit is called as? That point I will write here. So nucleic acids. So each monomer is, now let us write here. Nucleic acids means what? Many units are present. Like this. So in the case of nucleic acid, what the each unit is called as? Each monomer is called as nucleotide. Each unit is called as nucleotide. Observe carefully. Next one. Proteins are also polymers. Here also many units are present like this. And what this each unit is called? Each monomer is called as amino acid. Please observe the content carefully. Next one. Polysaccharides. These are also having many units. In the case of polysaccharide, what this each monomer is called as? Monosaccharide. But what about the lipids? They are not polymers. Very, very important. Observe carefully. They are not polymers. So here there is no concept of monomer. Please observe carefully. So in this way, what is the sentence we are saying? Each monomer of nucleic acid is nucleotide. Each monomer of protein is amino acid. Each monomer of polysaccharide is monosaccharide. Like this you can say. Reverse. Nucleotides attach to each other and they will form nucleic acids. Amino acids will attach to each other and they will form proteins. Monosaccharides will attach to each other and they will form polysaccharides. In this way also you can say. Yes or no? So next one. Next point, another thing you can say, nucleic acids are polymers of nucleotides, means many units of nucleotides will attach. Proteins are polymers of amino acids, you can say. Polysaccharides are polymers of monosaccharides, you can say. In this way, you can say. Reverse, if you are saying nucleotides polymerize and form nucleic acids, amino acids polymerize and form proteins, monosaccharides polymerize and form polysaccharides. Same information in any way you can say it, no problem. But this is what the overall concept is. And here if you see nucleic acids, proteins, polysaccharides, these are the original polymers. These three are polymers. These are the polymers. That's why they are true macromolecules. They are true macromolecules. True macromolecules. And their size is more than 10,000 kilodaltons. 10,000 kilodaltons. These three. These three. Now the major problem is with lipids. Lipids. Are they polymers? No, they are not polymers. And next point, they are not polymers. First point completed. Next one. What is the size of each lipid? Size of each lipid is less than 800 Dalton. Please observe carefully. Size of each lipid is again less than 800 Dalton. If it is less than 800 Daltons, where you are expecting this? In the acid soluble pool it should come, yes or no? If its size is less than 800 Daltons, it should come in acid soluble pool. But the problem is, where we are observing the lipids? In acid insoluble pool we are getting. Why we are getting in acid insoluble pool? This is the most important question. Many students, they know that lipids are present in this, but why they are present in this? They don't know the reason. So please listen very carefully. This is the very important point. Now in the beginning itself I told you, trichloroacetic acid is the solvent that we used here. Trichloroacetic acid is the solvent that we used here. And this solvent has polar nature. Polar nature. That means it is water soluble. But here we are talking about the lipids. Now let us see the nature of lipids and then we will see the explanation for this. Now we are talking about the lipids. Observe carefully. Lipids. Lipids are not soluble in water. Lipids are not soluble in water. And what is the nature of these lipids? They are non-polar. Lipids are non-polar in nature. Nature of lipids is non-polar. But trichloroacetic acid, the solvent that we used for this experiment is trichloroacetic acid. And what is the nature of this? It is a polar, means it is water soluble. It is polar, so it is water soluble. Observe carefully. Lipids are non-polar and TCA is polar. And now what is the problem here? I will give you some simple example first, then I will come to this original content. Let us take a glass, and in this glass let us take the water. Now on this water, let us add some oil. Now what happens? Observe carefully here. First let us add a drop of oil. Yes or no? Now, is oil miscible with water? Can oil mix with the water? No, it is not mixing with water. And that too, oil has less density, that's why it is floating on the water. Yes or no? This is one drop. Now let us add another drop here. So at a distance we are keeping it, fine. And then we will add one more drop. And slowly what happens? All these oil molecules, all these oil droplets, they will come nearer to each other and they will attach with each other. So that finally what happens? They are grouped together. Please observe carefully here. In the oil, when we are adding this oil, it is not mixed with water, and instead all these oil droplets, they are coming nearer to each other and they are attached to each other, and now they are forming a large, this point you please understand. Individual drop is small one, but after attachment what happens? Now they are making a large droplet. Yes or no? That's what happening here. Similar type of concept, you just use it here. Now We took a beaker here. In this beaker, we have added trichloroacetic acid. Trichloroacetic acid. This is the trichloroacetic acid. Observe carefully. Now what happens? In this already we have kept the tissue and we are breaking the tissue. And when we are breaking the tissue, what happens? So already in the cell chapter we studied, in the plasma membrane lipids are present. How they are present? They are present together. And now what happens? This membrane will rupture and lipids will come out. Okay, membrane will rupture, lipids will come out. But those lipids are not mixed with this trichloroacetic acid. So now what happens? All the lipids will aggregate together. Now observe the diagram carefully. All the lipids will aggregate together. All the lipids will aggregate together. They may form like this or they may form bilayer also. So in this way they are forming some vesicles. They are forming some vesicles. They may be unilayer vesicles like this or they may be bilayer vesicles like this. Okay, already we studied about this concept, right? So bilayer vesicles. Okay, so if they are forming the unilayer, generally we are using a word called as a micellis. Unilayer micellis, bilayer vesicles. So any of these words, and these words are interchangeable, so you can use them randomly also, so no problem. So in this way they may form unilayer or bilayer, whatever it is. If you see individual lipid, this is one lipid. If you see individual lipid, its size is less than, individual lipid size is less than 800 Daltons. But the solvent we used is a polar solvent. So since it is polar solvent, lipids are non-polar and these two are not mixed. And then what happens? These lipids are present together as micellis or the vesicles. And because of this, what happens? What is the size of this vesicle? Now it is more than 1000 Daltons. What is the size of this particular one? More than 1000 Daltons. Understood or not? So now what is the problem? For that cheesecloth, if you are observing here, this size is small. But since they are forming a large group like this, now it cannot pass through that small pore of the cheesecloth. Understood or not? Individual lipid is having less size. But the solvent we used is polar solvent and lipids are non-polar in nature. Because of this, they are forming the vesicles or the micellis that they are large in size. Since they are forming the large micellis or vesicles, they cannot pass through this small pore of the cheesecloth, and that is the reason why they are present along with this particular retentate. But originally they are small molecules. Due to the solvent nature, now they are forming aggregates, and now overall they are making the large size lipids and they are coming along with the retentate. This is the only reason. This is what the reason. So now for this particular thing, what is the reason here? The solvent that we used. The solvent that we used is polar solvent. Observe carefully. The solvent we used is polar solvent. That's why we are getting lipids in non-acid insoluble fraction. That is the retentate fraction. But originally they are not macromolecules. So that's why in NCERT very clearly they said lipids are not strictly macromolecules. They are not strictly macromolecules. They are not true macromolecules. Because of the solvent nature, they are coming into this particular fraction. That's it. This is what the overall concept that they are saying here. I hope now you understood this. And then now they may ask one application question. What is that application question? Instead of trichloroacetic acid, listen the question carefully. Instead of trichloroacetic acid, in this experiment, instead of trichloroacetic acid, suppose you are using non-polar solvent. Suppose you are using non-polar solvent. Please remember here, this is if condition. This is just an imaginary condition. If non-polar solvent is used in this experiment, then where you will expect these lipids? In the soluble fraction or insoluble fraction? Please tell me now. If non-polar solvent is used, where you can expect this particular lipids? In the soluble fraction or insoluble fraction? This is simple logic. So if you are using non-polar, lipids are also non-polar. non-polar will be miscible. So that's why if non-polar solvent is used in this experiment, lipids also will come along with the solvent. Lipids also will come along with the solvent. That means in the filtrate itself you will expect it. So but in this original experiment, why we are getting in the retentate? Because we used polar solvent. This is the technical logic that you need to understand. Clear? This is what the basic information. I hope now you understood this. With this particular information is completed. In this way, first we need to observe the fractions, filtrate, retentate. And again we are going with some chemical experiments. Experiments we need to do. Based on the experiments we can understand what are the organic compounds that are present, what are the inorganic compounds that are present. And in those compounds also what are the functional groups present, whether they are amino acids or proteins or lipids or nucleic acids or nucleotides, what exactly it is. Those things we can understand only based on the experiments. That's it. In this way, fractionation we are doing, and then we are going with the chemical experiments, and finally we can understand what are the various compounds that are present in the living tissue. And now you can get the complete list of organic and inorganic. And here they are saying that how to get this organic and inorganic compounds. Suppose you want to go for the analysis of inorganic compounds. Analysis of organic compounds is a little bit easy. But if you want list of inorganic compounds, from living tissue. Now they are talking about another experiment. Now observe carefully. You want only inorganic compounds. You don't want organic compounds. Now how you will do this? For this, in the NCERT very clearly they have mentioned, here we are using some destructive experiment. Whatever the tissue that we are taking, we need to burn the tissue. But what is the procedure here? Are you directly burning this? No. You need to go for burning, but what is the procedure here? First, let us take a fresh tissue. Fresh tissue means it contains water. So, if you are weighing this, whatever the weight that you are getting for this fresh tissue that is containing water, that weight is called as wet weight. Please observe carefully. If you are taking a fresh tissue, it contains water, and if you are getting the weight of this, that weight is called as wet weight because it is having the water. So now what we need to do? You need to dry this. Tissue. You just dry it. Because of drying, what happens? Water evaporates. Understood or not? Once water evaporates, now what you will get? Now you will get the dry tissue. From this water got evaporated. And if you are measuring the weight here, now this weight is called as dry weight. So definitely majority of the content of tissue is water only. Approximately 70 to 80% water is present. So if that water is evaporated, now this weight will decrease. Yes or no? Definitely wet weight is more because more water is present. If the entire water is evaporated, now whatever we are getting is called as dry weight. Definitely dry weight will be lesser than wet weight. And once you are getting this dry tissue, now you need to burn this. So because of the burning, what happens? Oxidation. Burning means what? So burning in the presence of oxygen. So here oxidation of compounds will occur. So oxidation of carbon compounds will occur. And how they are oxidized? They are oxidized and they are removed in the form of carbon dioxide and water vapor. So please remember here, your aim is, what is your aim? Your aim is to get inorganic compounds. Your aim is to get only inorganic. Only inorganic means organic compound should be removed. How you are removing this? First you are drying the tissue and then you are burning. Because of the burning, what happens? All the carbon compounds will be evaporated in the form of, means they are removed in the form of various gases like CO2, water vapor, etc. That means what happens? From the tissue, entire carbon compounds were removed. And once they are removed, what will remain? What will remain? That remain is called as ash powder. That white powder we are generally getting, right? White or some brownish powder or some grayish powder generally we are getting, that is called as ash. This is what. Understood or not? Once again, let me repeat here. Our aim is to get only an organic compound. So you need to remove organic compound. So generally, if you are taking a fresh tissue, it is having more water so that it is having more wet weight. So upon drying, what happens? Water will be evaporated so that we will get the dry weight, dry tissue. And this dry tissue will be burnt. It will be burnt in the presence of oxygen so that all the carbon compounds will be removed in the form of various gases. And then whatever the white powder or the grayish powder that we are getting, that is called as ash. So we are getting the ash. And what actually technically this ash contains? This ash contains inorganic compounds. This ash contains inorganic compounds. And now anyhow you know how to do the analysis using various chemical experiments. Using various chemical experiments, we will do the analysis of this particular ash. So you will obtain different types of inorganic compounds, only inorganic because organic were removed. So in this way, first we can prepare the ash, and upon analysis of this, we can identify what are the various inorganic compounds that are present in a living tissue. In this way, for the organic compounds, various other chemical techniques are present. You don't need to do like this. So with the help of that, we can understand the complete list of this particular organic compounds. So in the organic compounds also, generally what we do, so first we are going for fractionation. So from the big list of compounds, so individual fractions we will separate. So some few compounds will come in this, few will come in this, few will come in this. Again go for individual fractionation until you are getting each compound separately. Once you are getting each compound separately, then we will go for different types of experiments, and finally we will decide what are those compounds. This is what generally we are doing in the chemistry, and this is how we are obtaining the complete list of all organic compounds, inorganic compounds, ions, everything that is present in the living tissue. Understood? Not. Yes, this is what we are doing. Generally in the chemistry, what we are doing, what are the functional groups? Mainly we are focusing on the functional groups like aldehyde, ketone, and then amide groups, and then carboxylic groups. Like what are the various groups that are present? In the chemistry point of view, we are observing the various groups. But in the biology, we are not focusing on the functional groups. In the biology, we are classifying organic compounds into amino acids, proteins, and then sugars, and then nucleotides, nucleic acids. Likewise, we are classifying according to our requirement. That's it. In this way, we can get the complete chemical analysis for a living tissue. So in this way, we are getting the complete list. With this concept is completed. Before reading this NCERT content, there is another sentence in NCERT that I will try to tell you. Suppose you are talking about a cell. Cell. This is the cytoplasm. Cytoplasm. Not only cytoplasm, there are some organelles that are present. So these are the organelles. These are the organelles. Yes or no? So from all these things, overall, we are getting both organic and inorganic compounds. Organic and inorganic compounds. And in the organic also, organic large molecules, organic small molecules. Yes or no? This is what we are getting. And out of this, small organic and inorganic, all these we are getting in acid soluble pool. Acid soluble pool or acid soluble fraction. But the large organic molecules we are getting in acid insoluble pool. Acid insoluble pool. Okay. This is what with the original concept already we studied. But in NCERT, they have used a sentence. What they are saying? So this cytoplasm is actually mainly, not completely, mainly acid soluble pool. Acid soluble pool mainly represents cytoplasm. But please remember, even in the cytoplasm also, some proteins are present, some carbohydrates are present. So they will again come in acid insoluble fraction. Not just for cytoplasm, even in the organelles also, inside the organelles also some compounds are present. If they are small molecules, they will come under acid soluble pool. If they are large molecules, they will come under acid insoluble pool. So in this way, from the cell, wherever the small molecules are present, small molecules will come under acid soluble pool, whether they are from cytoplasm or from the organelles, wherever it is, they will come under acid soluble pool. If they are large molecules, they may be from cytoplasm, or they may be from the organelles, or they may be from the plasma membrane, wherever it is. So they all will come under acid insoluble pool. So simply based on the size, they are separated. But please remember, lipids, size wise, they are small molecules. But the solvent we used is trichloroacetic acid, which is a polar solvent. That is the reason why they are coming along with acid insoluble pool. But originally they are not true macromolecules. This is the overall content that they have given. With this chemical analysis part is completed. Now we are going with NCERT reading part. So we are reading this is the complete continuation information, but in NCERT in different paragraphs they give, we will read all those paragraphs a little bit quickly. Clear? Now let us quickly go through this. How to analyze the chemical composition? We can continue asking in the same way that what type of organic compounds are found in the living organisms? What type of organic compounds are present in the living organisms? How we can perform the experiment? Let us see. How does one go about finding the answer? So how you will give the answer for this one? There is an experimental procedure. To get an answer, one has to perform chemical analysis. To get this list of organic compounds, we need to perform chemical analysis. So how we are doing this chemical analysis? We can take any living tissue, we can take it. It may be a vegetable or a piece of liver or anything you can take. Any living tissue you can take it and grind it in trichloroacetic acid. This is the Cl3CCOOH, or simply you can write CCl3COOH. Like this you can easily pronounce this. And this is a polar solvent. Underline trichloroacetic acid and write that it is a polar solvent. And using this particular solvent and using mortar and a pestle. So mortar and a pestle, you can grind it. Once you are grinding, what happens? Cells will break and cellular content is released into this trichloroacetic acid, so that we obtain a thick slurry. Slurry means it is a semi-solid paste. Slurry is semi-solid paste we will get. Next one, if we were to strain this through a cheesecloth, so this slurry we are straining, passing through a cheesecloth or cotton, then we would obtain two fractions. We will get two fractions. What are these? One is called as a filtrate. Inside the beaker we are getting, no, that is the filtrate, or more technically it is called as acid soluble pool. This is the one fraction. Then what is the other fraction? And the second fraction is called as the retentate because it is present above the cheesecloth. It is also called as acid insoluble fraction. In this way, two fractions we are getting. Scientists have found thousands of organic compounds in the acid soluble pool. In the acid soluble pool, thousands of organic compounds were identified. But in the acid insoluble pool, 4 types of organic compounds. Please remember this. In the acid insoluble pool, just 4 types. But in the acid soluble, means small molecules, thousands of small molecules are present in this. Okay, for the organic. Not only that, even inorganic are also present in this. Next one, in higher classes, you will learn about how to analyze a living tissue sample and identify particular organic compound. So at the level of class 11th and 12th, we are not studying this, but in higher classes you will study different techniques that are used to isolate the compound and identify the compound. Many techniques are there. So those you will study in higher classes. It will suffice to say here that here it is sufficient to say that one extracts the compounds, then subject the extract to various separation techniques till one has separated a compound from all other compounds. Here we are not worried about the complete experiment, but it is enough. What is enough? We just understand that initially group of compounds are present. So we are separating so that each compound is separated. Like that we are doing the separation. If you understand this, that is enough. That's what they are saying. Next one, in other words, one isolates and purifies a compound. That means from the group of compounds we are isolating it and then we are purifying it. Purifying means only one type of compound is there. Other type of compounds are not present. So in this way we are isolating and purifying the compounds from that initial group. And analytical techniques means what? For identification of this we are using these techniques. When analytical techniques, when applied to the compound, they will give us an idea of molecular formula and the probable structure of the compound. Using various techniques we can identify the compound and even we can get the structure of compound also. That's what they are saying. Next one, all the carbon compounds that we get from living tissues can be called as biomolecules. So what are these biomolecules? Molecules in living systems. So that's why biomolecules. So that's why not just here they are saying that all the carbon compounds, not just carbon compounds, all the compounds from the living tissues, you can call them as biomolecules. In simple. Next one. However, living organisms have also got inorganic elements, not only organic compounds. In the living organisms, even inorganic compounds are also present. Inorganic elements are also present. Inorganic compounds are also present. Okay. Next one. How do we know this? How do we know about these inorganic compounds? If you want to get the information of inorganic compounds, a slightly different but destructive experiment, because you are burning it. Once you are burning, you cannot get it back. Yes or no? That's why it is a destructive experiment has to be done. For what? To get inorganic compounds list. Not for organic. For inorganic compounds list, we are doing this. Next one. One weighs a small amount of living tissue. So let us take a small amount of living tissue. Say it may be a leaf from a plant or liver from an animal, and it is called as a wet weight. Why it is called wet weight? Because it is a fresh tissue. It is having more water in this. That's why it is having wet weight. And this tissue, you are drying it. Upon drying, what happens? Water will be evaporated. You will get the dry weight. And all the water will evaporate. Because of the drying, what happens? Water will be evaporated. And the remaining material gives, after evaporation of the water, the remaining material will give dry weight. Yes. Next one. Now, if the tissue is fully burned, suppose you are burning this tissue now, what happens? All the carbon compounds are oxidized to some gases like CO2 and water. Carbon compounds are removed in the form of some gases and they are removed. What is remaining is called as ash. After removal of this carbon compounds, some powder we are getting, and that remaining is called as ash. This ash contains inorganic elements and compounds. In this ash, we are getting the inorganic compounds and elements. Do the experiments, you will get the complete list. That's what they are saying. Inorganic elements like calcium, magnesium are present. And inorganic compounds are also present, like sulfates are present, phosphates are present, carbonates are present, many are present, are also seen in acid soluble fraction. Please understand this. Now, this is what you are doing. And now these inorganic, where we are getting? In the acid soluble, not in the insoluble. Please remember, in the filtrate we are getting inorganic. So in the filtrate what we are getting? Small organic compounds and all the inorganic compounds we are getting in acid soluble fraction. In the acid insoluble fraction, only 4 types of organic compounds we are getting. So please observe this. Many students are ignoring these contents. So please remember, each and every point is important for examination. In this way, all the inorganic compounds we are getting in acid soluble fraction or acid soluble pool, POOL pool. Next one. Therefore, elemental analysis gives elemental composition of living tissues in the form of hydrogen, oxygen, chlorine, carbon, etc. If you are doing elemental analysis, you will get the complete list of elements. If you are doing the compound analysis, what are the various compounds that are present, that list you will get it. While analysis for, here we are doing elemental analysis, so we are getting the elements. While analysis for compounds gives us an idea of the kind of organic and inorganic constituents present in the living tissues, not in the ash. Ash will have only inorganic, but here for living tissue they are saying. If you are doing the compound analysis, you will get what are the various organic compounds present, what are various inorganic compounds present. If you are doing elemental analysis, what are the elements that are present? That list you will get. So in this way, complete list you can get here. Okay, next one. From a chemistry point of view, one can identify functional groups like aldehydes, ketones, aromatic compounds, etc. Mainly in the chemistry, they will focus on what are the functional groups that are present. But from a biological point of view, as a biologist, we are not focusing on just functional groups. As a whole molecule, we are classifying and then we are separating them. So in the biological point of view, we shall classify them into amino acids, nucleotide bases, fatty acids, sugars. Likewise, we are classifying. In this way, according to our requirement, we are classifying like this. If you are observing the inorganic compounds list, so what are the inorganic compounds? Sodium present, potassium present, calcium, magnesium, water present, sodium chloride, calcium carbonate, phosphates, sulfates, likewise many are present. In this way, ash analysis, from the ash analysis, we can get various list of inorganic compounds. This is what they have given. Next, and there it is completed. And after this paragraph in the NCERT, they are talking about amino acids. After that, they are talking about lipids. After that, they are talking about nucleotides and all. Likewise, they have gone. And after that, separately, they have given this particular paragraph. Separately, bio macromolecules. Here also again they are talking about filtrate, retentate, micromolecule, macromolecule, size, everything they are talking. So once again it is related to the same experiment. So now let us see again. There is one feature common to all those compounds found in the acid soluble pool. Those compounds that are present in acid soluble pool, for them there is a common feature. What is that common feature for acid soluble pool? They have molecular weight ranging from 18 to 800 Daltons approximately. Their size is small and their range is between 18 Daltons to 800 Daltons. That means less than 1000 Daltons. In this way they are present. So in the acid soluble pool, all the molecules are small molecules. That's why they are called micromolecules. Next one, the acid insoluble. Insoluble means retentate. Insoluble fraction has only 4 types of organic compounds. Only organic compounds are there. That too only 4 types are there. What are those? Proteins, nucleic acids, polysaccharides and lipids. These are the 4 types of organic compounds that are present. Next one, these classes of compounds with the exception of lipids, underline this. Out of these 4, you just remove the lipids. Why you are removing the lipids and what they are saying? First we will read, then we will see. With the exception of lipids, have molecular weights range, or we have molecular weights in the range of 10,000 Daltons and above. So who is having this? Proteins, polysaccharides, nucleic acids. Only these 3 are having more than 10,000 Daltons. But lipids, they are not having more size. Please remember this. Even though lipids are present along with this particular fraction, they are not true macromolecules. Only these 3 are macromolecules. Only these 3 are polymers. Only these 3 are having size more than 10,000 Daltons. That's what they are saying. And for this very reason, biomolecules, that is chemical compounds from living organisms, biomolecules, molecules from biological living organisms. So compounds found in living organisms are of 2 types. So that's why overall, if you are observing, we are classifying them into 2 types. One, what is the first one? Those which have molecular weights less than 1000 Dalton, less than 1000 or less than 800 Dalton, whatever it is, and are usually referred to as micromolecules. Understood or not? So in the acid soluble pool, small molecules are obtained and they are called as micromolecules, or simply they are called as biomolecules, or simply they are called as biomolecules. Biomolecules means not only small molecules, even large molecules also will come, but generally they are saying that this acid soluble fraction biomolecules they are saying. Why they are saying like this? Because see, amino acids are coming in acid soluble pool, but their polymer proteins will come in acid insoluble. Individual nucleotides will come in acid soluble pool, but nucleic acid will come in acid insoluble pool. Individual monosaccharides will come in acid soluble pool, but polysaccharides will come in acid insoluble pool. But ultimately, they are making these things. So that means overall, this acid soluble pool will represent this biomolecules. But not only this, insoluble fraction also will come under biomolecules. While Those which are found in acid insoluble fraction are called as macromolecules or biomacromolecules. So in this way, two fractions: acid soluble pool, biomicromolecules; acid insoluble pool, biomacromolecules. This is what they are saying. Next paragraph. The molecules in the insoluble fraction, with the exception of lipids, every time they are very clearly saying with the exception of lipids, are polymeric substance. That means lipids are not polymers. Nucleic acids are polymers, proteins are polymers, polysaccharides are polymers. So these three are polymers, but lipids are not polymers. Next one. Then now they are asking question. If lipids are not polymers, then why do lipids whose molecular weights do not exceed 800 Dalton? Each lipid less than 800 Dalton. But still, why do these lipids come under acid insoluble fraction? That is macromolecular fraction. This is the most important question. Individual lipid is less than 800 Dalton. We are expecting that it should come under acid soluble pool, but originally it is coming in acid insoluble pool along with macromolecules. Why it is coming like that? That's what they are saying. Now what is the answer they have given? Lipids are indeed small molecular weight compounds. Lipids are actually small molecular weight compounds and are present not only as such but also arranged into structures like cell membrane and other membranes. Membranes means what? These lipids are aggregated together so that they are forming some membranes, vesicles, and then micelles. Likewise, they are forming this. Yes or no? In this way, they are forming. So they are not present as such. They are not only present as such. That means some lipids are present individually, but majority of these lipids are present together in the form of membranes or some other vesicles. When we grind a tissue, that means when we are breaking a tissue, we are disrupting the cell structure. So we are breaking the cell structure. Disrupting means breaking the cell structure. Then cell membrane and other membranes are broken into pieces. So at that time, plasma membrane and the membranes of organelles are broken into small fragments, pieces, and they will form vesicles which are not water soluble. Please remember here. So vesicles they are forming, but these vesicles are not water soluble. The solvent we used is water soluble solvent, polar solvent we used, but lipids are non-polar. They are not soluble in water. This is the problem, and these are not water soluble. Therefore, these membrane fragments in the form of vesicles get separated along with acid insoluble pool, and hence the macromolecular fraction. So since these lipids are not soluble in water, they are not going with the acid. They are present along with macromolecules. Since they are grouped together, their size, overall size is increased now. So they are not passing through the small pores of the cheesecloth. So that's why they are present in acid insoluble pool. That's why they are coming along with macromolecular fraction. Lipids are not strictly macromolecules. They are not originally macromolecules. Only the remaining three are macromolecules. Lipids are not true macromolecules. Next paragraph. The acid soluble pool represents. So what is present in the acid soluble pool? It is represents roughly the cytoplasmic composition. Why they are saying roughly cytoplasmic? Because even in the cytoplasm also some proteins and carbohydrates are present, and they will go into acid insoluble. So that's why acid soluble pool roughly represents cytoplasm. Next one, the macromolecules from the cytoplasm and from the organelles, all of them, they will become acid insoluble fraction. Wherever macromolecules are there, all the macromolecules will go into acid insoluble fraction. Next one, together they represent the entire chemical composition of living tissue or organism. So acid soluble, acid insoluble combinedly, now they will represent the composition of a cell or composition of a living tissue or composition of living organism. That's what they are saying. Clear? Next one. In summary, if we represent the chemical composition of living tissue from abundance point of view, that means suppose, so after the analysis we are classifying all the compounds, and who is more abundant, who is less abundant. That means who is more present, who is less present. Based on the abundance wise, if you are arranging this, and again on the class wise if we are arranging them, then we observe that water is the most abundant chemical in the organism. So what is the percentage wise if you see? Water 70 to 90%, proteins 10 to 15%, carbohydrates 3%, lipids 2%, nucleic acids 5 to 7%, ions 1%. So overall, whether they are see, water is not organic compound. If you want to say as compound, so this will come under inorganic. Okay. So now if you are arranging all of them, now water is the most abundant. After that, proteins will come. After that, nucleic acids will come. After that, carbohydrates will come. After that, lipids will come. And finally, ions will come. This is the sequence. This is the overall sequence for all organic and inorganic combined. Please remember the question here. If they are asking only about the organic compounds, if they are asking only about organic, you should not talk about water. You should not talk about ions. Please remember. In the sequence, in general, they are asking water greater than proteins, greater than nucleic acids, greater than carbohydrates, greater than lipids, greater than ions. This is the sequence. Suppose they are asking only organic compounds. You should not talk about water, you should not talk about ions. Then what is the sequence? Proteins greater than nucleic acids, greater than carbohydrates, greater than lipids. This is the sequence. So most abundant organic compound if they ask, proteins. Most abundant organic compounds are the proteins. Yes or no? So overall if they ask for the cell, it will be the water. Most abundant compound simply they are asking, then it will be the water. In this way also questions can be asked. So with this chemical analysis for a living tissue is completed. And now what we will do is, in the next lecture onwards, we will go with individual components. What we will do? We will go with amino acids first. After completion of this, we will go with proteins. Then we will go with nucleotides, structures, everything we need to study. So please remember. After that, we will go with nucleic acids. And then we will go with lipids. Finally, we will go with monosaccharides, some disaccharides, and then some polysaccharides we will study. If we study this, majority of this chapter is completed. And then some information related to some enzymes and some other information they have given. Those things we can go with. Clear? So in this way we are going. Please remember, ultimately we are completing this chapter from NCERT, but we are studying it in a proper way, so that you can easily understand this particular information from this particular chapter. Okay. So in the next lecture, most probably we will start with amino acids. Okay. We will meet in the next lecture. Thank you so much.